Hello everyone, welcome to our YouTube channel Unfog with Dr. Atar Parveen. In today's video, we'll be discussing the remaining 20 questions of the 2015 Hstr Biology question paper. Please subscribe our channel and like our video, and please share this video as much as possible with CB method aspirants for more such videos. So, let's begin with our class. The 61st question says, match the following with the cells of phloem tissues in column 1 with their functions in the column 2. Identify the correct choice. So we have to match the terms which are given in column 1 with their correct functions which is given in the column 2. So let's find out. In column 1, the first one A is the sieve elements. So what are sieve elements? Sieve elements, these are elongated living cells of phloem which help in food transport from the leaves to the entire plant. Okay. So which of the options that is matching? The correct function that matches our definition is option number P. So option P is the right answer for sieve elements. Now companion cells. Now companion cells, these are type of cells found in the phloem and help in loading and unloading sugar into the sieve elements. So basically they help in the transportation and control of food materials. Okay. So the correct option from the column 2 that matches our definition is option number T. So for companion cells, it is T. Now C, parenchyma. So parenchyma cells, these form the bulk of the plant ground tissues. Means the majority of the cells that are present in the plant, they are actually parenchyma cells. And these parenchyma cells are living cells and they perform functions like photosynthesis, storage, transport, and the correct function that is matching with our definition from column 2 is the option number R. So for parenchyma cells, the option is R. Now last one we have is fibers. So what are fibers? Fibers are greatly elongated cells whose long tapering ends interlock. Okay. So if I show fibers, they kind of look like this. Okay. So these ends, these that are present here, they are known as the tapering ends. So different fibers, how they are aligned means, this is first fiber, the other fiber is present like this. Means the first end and the last end of the first fiber tissue, they are present very close to each other. Means interlocked with each other. So what happens when they are interlocked with each other is that they provide maximum mechanical support to the plant. Mechanical support in the sense, how is the plant standing straight, right? How it is standing so straight even when the wind is blowing so fast, or even in sometimes when there is a flood, trees stand strong, right? So what are the type of tissues that are helping it stand so steadily? They are the fiber cells. And the correct function that matches our definition from column 2 is the option S. So the correct answer for fibers is option S, which says elongated cells provide mechanical support. Now let's see which one of these options match with our correct sequence. So option A is P. Okay, all are having P here. Then we'll have to look for B. B is given in first and third option. B for T, right? So first and third option is given. So we'll cancel out second option and the fourth option. Now we have C. For C we have R. And that R is present only in third option. So third option must be correct. So if we want to cross check, we'll rectify our last option also. For D we have the S, right? So the third option is the right answer. The next question is, the nucleosides of DNA are. The first option is deoxyribose sugar plus water. Second one is deoxyribose sugar plus nitrogenous bases. Third option is deoxyribose sugar plus nitrogenous bases plus phosphate group. Fourth option is deoxyribose sugar plus nitrogenous bases plus phosphate group plus water. So what is a nucleoside first of all? So nucleoside, it is an organic molecule with a basic composition of a nitrogenous base, pentose sugar and a phosphate. So nucleoside looks like this. This is a pentose sugar. Pentose sugar means it has a 5 carbons in it. Okay. So we have 1, 2, 3, 4 and 5. So pentose sugar means it is a 5 carbon sugar. And this nucleotide has one sugar and one nitrogenous base which is joined to the second carbon. We'll show this nitrogenous base with N, and a phosphate group which is present on the fifth carbon, and we'll show it with P. Okay. So nucleosides are made up of, we'll write it in a formula. Nucleosides are made up of a pentose sugar. Pentose sugar means it has 5 carbons in it, and one nitrogenous base. That nitrogenous base can be anything, like we have adenine, guanine, cytosine, thymine, and in plants we have uracil, right? So nitrogenous bases plus a phosphate group. Let's see which one of the options is matching with our answer. So nucleoside means pentose sugar. Okay, we have written pentose sugar here. They have given deoxyribose sugar in the options. They are the same thing. So we have deoxyribose sugar plus nitrogenous bases. Okay, here water is present, so this must be the wrong answer. Here only nitrogenous bases is present. This is also the wrong answer. Then we have deoxyribose sugar, nitrogenous bases and phosphate group. So third option is the right answer. The fourth option is automatically wrong. We don't have to look at it. So the third option is the right answer. Next question. If an individual is heterozygous for both characters as in F1 in Mendelian dihybrid inheritance, is test crossed with double recessive, the phenotypic ratio obtained is. Okay, it sounds a little bit scary, right? But it is very simple. Let me tell you how. If an individual is heterozygous means it has two different alleles for one trait. For example, we'll take capital A is for tall and small a is for dwarf. Okay. So capital A, small a, because we are representing tall with capital A, it means it is a dominant character. And he has given it is a heterozygous individual. Heterozygous means there should be one capital A and there should be one small a. So one capital A and small a together will make a heterozygous individual. Okay. Now we are saying that tall, we are representing it with capital A. So it is a dominant character. And small a, we are representing that with dwarf. So that automatically becomes the recessive characters. And the recessive characters are always suppressed by the dominant character. So now here one dominant character is present, one recessive character is present. So the recessive is suppressed by the dominant, and heterozygous tall we will have here. This individual will be heterozygous tall. Now he is saying for both characters, means only height is not one character. There is some other character also present. It could be any characters, but for our understanding we are taking simple characters here. Okay. So there are two characters. First character we took in consideration here is height. Okay. Now we'll take another character into consideration. For example, let's take flower color. Okay. So we have flower color, which is the second character, and we'll represent it with capital B and a small b. Capital B means we are taking a capital letter here, so it becomes a dominant character. So that we'll take it as a white color. So white color, it is the dominant character color, and we are taking small b here, so it becomes the recessive character. And let's take this recessive as yellow color. Okay, so now he's saying that individual is heterozygous. So heterozygous means there should be one capital and one small b, right? So this is again heterozygous. When we put both of these characters together, it becomes capital A, small a, capital B, small b. Because here in question he is referring, no, heterozygous for both characters, right? So we'll have to take height also and flower color also. So this is our individual, main individual here, who is having tall height and is having white flowers, because this capital B dominant character will dominate the recessive character. So it is a tall plant with white colored flowers, and this is the F1 generation according to the question. See, as in F1 in Mendelian dihybrid inheritance. See, he is saying dihybrid. Dihybrid means we take two characters in that cross, right? And this F1, it is crossed, test crossed with double recessive. Okay, so basically there are two types of crosses. One is back cross, one is test cross. Back cross means that progeny, that F1 generation that you are getting, we cross it with one of its parents. That is known as the back cross. But if we are crossing this F1 generation with a double recessive individual. Now double recessive means, see, there is two terms here, double and recessive. Recessive means it will be small letters. Double means they are twice present, right? So we'll denote it as small a, small b, small b. So this becomes the double recessive individual. Okay. So when we are crossing the F1 generation with a double recessive individual, it is known as a test cross. So now he is saying that we are crossing this F1 generation with the double recessive individual, right? So now he wants us to tell the phenotypic ratio. Phenotypic ratio means, phenotype is the characters that are observable and they are controlled by the genetic constitution of the organism. That is known as the phenotype, right? And we have two types of ratio, phenotypic ratio and genotypic ratio. Genotype is the genetic constitution of the organism, but he is only asking us of the phenotypic ratio, like what all type of plants will be present and in what ratio will they be present. Okay, so for that we have to do a dihybrid cross. First we'll have to form the gametes for that. Now for us this capital A, capital B, small b is the first parent, and small a, small b is the second parent. Let's take, this is a female, this is a male. So now we have to form the gametes first, right? So capital A, capital B, small b. In easy way you can form the gametes. I'll tell you a trick. Okay. So 4 gametes will form all in all. So first we'll form, write capital A, small a. Now next we'll write down small b, capital B, small b. Okay. So these are our gametes for the female parent. Now we have to form gametes for the male parent also. So we have a small b, small b. Now 4 gametes we need to form because they all are the same. we'll form it as small a small b, small a small b, small a small b, small a small b. Because there is only two varieties present. No, small a and small b. So similar type of 4 gametes will be formed. Now we have to do the cross. We'll draw a Punnett square for this. Okay, now we'll write down the female gametes first, which are capital A, small b, capital A, capital B, small a, capital B, small a, small b. And now here we'll write down the male characters. Okay, now let's differentiate them so we are not confused. So small a, small b, then again small a, small b, small a, small b, and again small a, small b. Now we'll have to make a cross between them. Okay, so capital A, small a, small b, small b. This is the first one. Okay, next we have is capital A, small a, capital B, small b. Then again we have small a, capital B, small b. Then again we have small a, small b, small b. Then next we have to cross with this character and this character. Capital A, small b, small b. Then again we have capital A, capital B, small b. Then again we have small a, small a. Then capital B, small b. You have to be very careful when we are doing dihybrid cross because you'll get confused. Okay, so see properly and write down the characters properly. Then next we have small a and small b, small b. Then we'll again cross with this capital A, small b, small b. Capital A, small b. Sorry, capital B here we have, right? Capital B, small b. Then small a, capital B, small b. Small a, small b, small b. Then next we have again capital A, small b, small b. Capital A, capital B, small b. Then again we have small a, capital B, small b. Small a, small b and small b. Okay. Now let's write down the phenotype. What all are we observing here? What type of plants, what type of progeny we are getting here? So in phenotype, we can see that there is a capital A, capital B, small b. Okay, we'll write down. So in capital A, small b, capital A, small a, capital B, small b. Now we have the dominant characters here. So they'll dominate the recessive character. And then we'll get capital A is for tall. And capital B is for white. So how many of such type are present? Capital A, capital B, small b. These 4 are present. 1, 2, 3 and 4. So 4 such types we are getting. Tall, white, flowered plant. Now next we have capital A, small b, small b. Okay, here both are recessive characters. Now remember, when recessive characters are in the homozygous form, homozygous means both the letters present are same, small b, small b. They are same letters, right? So if they are present when they are same, small b, or small a, in the homozygous form only the recessive character can be expressed. Okay, now we have capital A here and small a here. Capital A, of course, it will dominate again and will get tall. But here we have recessive character, but they are present in homozygous form. 2 similar characters are present. So we'll have yellow colored flowers. And this also, how many are present? Capital A, small b, 4 are present. Right? So 4 such plants will be obtained. Now next we have is small a, capital B, small b. Now that small a, capital B, small b. Now here small a is present in the homozygous recessive form. So small a is for dwarf. Okay. And next one is capital B, small b. Capital B is a dominant character. It will dominate the recessive character and we will get white colored flowers. Okay. And how many such plants are we getting? We are getting 4 such plants. Now next type we have is small a, small b. Right? Now small a, small b, they are both recessive, right? They are homozygous recessive. So we are getting dwarf yellow colored flowers wala plant. And how many such plants are present? 4 such plants are present. Now these are the total genotypes we have, and from those genotypes we made out the phenotype also. Now what is the ratio of these 4? It is 4:4:4:4. But this ratio should be represented in its simplest form, right? We can cancel out this 1, 4 ones are 4, 4 ones are 4, 4 ones are 4. So we'll cancel it out, right? So in its simplest form, it can be represented as 1:1:1:1:1. So the phenotypic ratio is 1:1:1:1. So which one of the options is matching? The second option is matching with our answer. So the second option is the right answer. Yes, I know this is a lengthy process and it is very sometimes confusing to make out this genotypes in the dihybrid cross, but it is not difficult. We can do it with practice and with concentration. It is one of the easiest and the scoring sums. Okay. So don't get scared of this. Try to make it out with a cool mind. And please be careful about what type of letter is present. It is a capital letter or a small letter, because the small letter or a capital letter will decide what is the phenotype and the genotype of the organism. So be careful when writing down the letters. And according to this, our answer should be 1:1:1:1 phenotypic ratio. Next question. The female reproductive part of Polysiphonia is called as option 1 Oogonium, option 2 Carpagonium, option 3 female conceptacle, and option 4 cryptoblast. The answer is option number 2. Carpagonium is the female reproductive part of the Polysiphonia. Next, the mulberry, tussar, and muga are the varieties of. Option 1 is given as leaves eaten by silkworms. Option 2 as cultivated silks using different silkworms. Option 3 natural silk. Or option 4 process of separating the silk fiber. Actually mulberry, tussar and muga, these are all the leaves, the different types of leaves which are eaten by the silkworms. So option 1 is the right answer. Next question. The bee species which is not possible to domesticate is. So domesticate in the sense like we have domestic animals, right? We have cows, sheep, goats, buffaloes, and sometimes even honeybees. We can cultivate honeybees also, right? It is known as apiculture. So which type of bee species from these 4 options cannot be domesticated? In the sense they are harmful also. Okay. So the first option is Apis cerana indica, option 2 Apis indica, option 3 Apis dorsata, and option 4 Apis mellifera. The right answer is option 3 Apis dorsata, and it is that bee species which cannot be domesticated. It is not possible. Next question. The connecting link between Annelida and Arthropoda is option 1 Lepidosiren, option 2 Ornithorhynchus, option 3 Peripatus, and option 4 Sphenodon. So option 3 is Peripatus is the connecting link between the Annelida and Arthropoda. Next question. The technique of DNA fingerprinting was developed by option 1 Carey Mullis, option 2 Maurice Wilkins, option 3 Baltimore D, and option 4 Alec Jeffreys. So it was actually option 4 Alec Jeffreys, scientist, who gave the technique of DNA fingerprinting. Next question is, Elasmobranchi is the group which includes option 1 lungfishes, option 2 catfishes, option 3 cartilaginous fishes, and option 4 bony fishes. So Elasmobranchi is that group of fishes which includes cartilaginous fishes. Cartilaginous fishes means they'll have bones which are made up of cartilage. And when I say bones made up of cartilage, it means that they'll have very flexible bodies. And fishes like sharks, rays, soft fishes, these all come under cartilaginous fishes, and these cartilaginous fishes are placed under the Elasmobranchi group. So cartilaginous fishes is the right answer. Please subscribe our channel and like our video, and please share this video as much as possible with CB method aspirants for more such videos. Next question. Read the statements A and B, indicate the correct choice from these given below. So we have two statements A and B here, and we have to tell either these two statements are true or false, or which one of them is true or other one is false. Okay. So first statement we have here is, insulin is a small protein. It consists of 51 amino acids in A and B chains. Statement A is true. Okay, so insulin, it is actually a protein. Proteins, when we say, it means that they are long chains which are made up of small units which are called as amino acids. So this amino acids, these small balls that I am drawing here, these are the amino acids. And when these amino acids, they are linked together by a peptide bond. This line here is known as the peptide bond. Okay, they form a protein. So when 51 such balls are present, it forms a insulin. Okay, now this insulin, actually it is divided into two chains, chain A and chain B. Here one other chain will be there, and this chain will be known as the chain B. And in this chain A, there are actually 21 amino acids present. So 21 amino acids means 21 such balls will be present. And in chain B, there are 30 amino acids present, and these two chains, they are linked together by disulfide bridges. Disulfide bridges means they'll form a sulfate linkage here. Here sulfur is present, right? So this two chains, they are linked together by a disulfide bond. Two sulfides. So di is present. Disulfide bond is present between these between these two chains, and these together will form a insulin. So let's read the statement B. A polypeptide chain which consists of 21 amino acids. Yes, 21 amino acids are present. And B polypeptide chain has 30 amino acids. Yes, 30 amino acids are present. And these chains are linked by a disulfide bond. That's what we studied, right? Here disulfide bonds are present. So statement B is also true. So let's see which one of the option is matching with our answer. Both the statements A and B are true. Option 3 is the right answer. Next question. Mode of nutrition in earthworm is option 1 Holozoic, option 2 Coprozoic, option 3 Holophytic, and option 4 Saprozoic. So the mode of nutrition in earthworm is Holozoic. So Holozoic means these organisms, they will surround the food material that is present. They'll ingest it and they'll digest it inside their bodies. Okay, so that is the Holozoic mode of nutrition which can be seen in the earthworms. Next, the culturing of undifferentiated cell mass Let's underline this. Undifferentiated cell mass which are initiated from an explant of seedling or other plant tissue source is option 1 callus culture, option 2 tissue culture, option 3 organ culture, and option 4 protoplast culture. If we look at these options, this callus culture, organ culture, protoplast culture, these are all subdivided parts of tissue culture only. So tissue culture means we can take any part of the plant or an animal and then we can culture it. We can grow it in a in vitro form, right? Outside the body in an artificial medium. That is known as the tissue culturing. So this tissue culture can be of callus culture, can be divided into different types. Like we have callus culture, then we have organ culture, then again we have the protoplast culture. Many such types of different tissue culturing are present. But here in options we have callus culture, organ culture and protoplast culture. So callus culture means it is that undifferentiated cell mass which is taken from the plant body and it is cultured. That is known as the callus culture. And that is what is being asked in the question. So actually callus culture is our right answer. In organ culture, what they'll do, they'll take any or part or the any organ of the plant and they'll culture it outside in vitro conditions, in artificial conditions. That is known as the organ culture. Now protoplast culturing means a cell without the cell membrane. It is known as the protoplast, and such type of content it is cultured in vitro conditions again. So that is known as the protoplast culture. But according to our question, the callus culture is the right answer. Next question. The technique which is carried out to confirm the presence of foreign DNA in the offspring is option 1 is Western blot, option 2 tail blot, option 3 ELISA, and option 4 PCR test. The correct answer is the PCR test. So what is this PCR test? That PCR is the short form for polymerase chain reaction. So this when put in short is called as PCR test. So what it does is it is used for amplification of DNA. Amplification means if there is one DNA segment present, then we can form hundreds and 500s and thousands of DNA similar segments by this test. So what it does, it will generate millions of copies of a small segment of DNA. Now we are saying that we want to find the presence of a foreign DNA in the offspring. Now this foreign DNA can even be in minute tiny amounts, right? So how will we come to know that if it is present in the offspring or not? We'll make multiple copies of this similar DNA by the PCR test and then we can observe that there is a foreign DNA present. So PCR test can be used to confirm the presence of foreign DNA in the offspring. Next question. When a cell expends energy to move a solute across its membrane against a concentration gradient, the process is called as option 1 diffusion, option 2 facilitated diffusion, option 3 active transport, and option 4 osmosis. So option 1 we are talking about diffusion. So diffusion means the solute particles will flow from their higher concentration to lower concentration without any need of any membrane. Like we have fully permeable membrane and semi-permeable membranes, right? So such membranes are not required for the movement of the particles, and it goes from the higher concentration gradient to lower concentration gradient. So again no energy will be required. So option first is wrong answer. Second one we have is facilitated diffusion. So facilitated diffusion means now we will need a membrane to help in transport of these solute particles. Now we have now for example let's say this is a cell membrane. Okay. Now in this cell membrane, there will be a protein channel present here. That protein channel is actually called as porin. This porin helps in the transport of molecules from the outside of the cell to the inside of the cell. Now for example, I have to transport for say water molecule itself. This is H2O. Okay, now this water molecule will go inside this protein channel, and this protein channel will let that water molecule inside the cell. Now this is facilitated diffusion because this transport of water molecule is being facilitated by this protein channel called porin, right? So this is a facilitated diffusion, and this also occurs from the higher concentration gradient. Higher concentration gradient means here the concentration outside the cell is high and inside the cell is low. So water flows from higher concentration to lower concentration, and again no energy will be required. So facilitated diffusion is also the wrong answer. In active transport, what happens is, now let's take, this is the same cell membrane. Now we have to transport the water molecule itself from outside the cell to inside the cell, but outside the concentration of water is low and inside the cell the concentration of water is high. For to doing that will require some sort of energy because we are pushing against the concentration gradient, right? And ATP will be required to push against the concentration gradient. So here energy is being expended, right? So active transport will be the right answer. Then we have osmosis. In osmosis also, there will be a semi-permeable membrane present, and that semi-permeable membrane through which water molecules will flow, but they'll flow from lower concentration to higher concentration only, but no energy will be required here. So osmosis is the wrong answer, and active transport automatically becomes the right answer. The next question we have is syphilis, which is a name of a disease, is caused by the virus. Option 1 we have Vibrio cholerae. So Vibrio cholerae, as the name suggests, it causes cholera. So this becomes the wrong answer. Next we have Neisseria gonorrhoeae. This Neisseria gonorrhoeae, as the name suggests, it causes gonorrhea. So again this is the wrong answer. Third one we have Clostridium botulinum. This Clostridium botulinum causes botulism. Okay, and in this botulism what happens is there is a muscle paralysis and all such paralysis occurs. So Clostridium botulinum causes botulism. This again is the wrong answer. Now remaining automatically we have the Treponema pallidum. So Treponema pallidum causes the syphilis. Next, the special channels found in the plasma membrane for water transport are. We discussed in the previous question, right? The water is molecule is present. It is transported through a protein channel called as a porin. Now we have specialized channels in the plasma membrane which help in the transport of water itself. Now water is also called as aqua, right? So the porin or the protein channel, special channels responsible for transport of this water will be called as aquaporins. So the second option, aquaporins, is the right answer. You don't even have to look at the remaining other options. Okay. So aquaporins will be our right answer. Next question. The cell organelle having half unit membrane is option 1 ribosome, option 2 spherosomes, option 3 lysosomes, and option 4 peroxisomes. So option 2 spherosomes are the cell organelles which have half unit membrane. Next question. The path which is often called as facultative pathway is option 1 C3 pathway, option 2 C4 pathway, option 3 CEAM pathway or also called as CAM pathway, and option 4 C2 pathway. It is the CAM pathway which is often referred to as the facultative pathway. Next question. In glycolysis, ATP is synthesized by. So we need to know the glycolysis process, and ATP is synthesized by. The first option is fructose 1,6 biphosphate. Option 2, phosphofructokinase 1. Option 3, glyceraldehyde 3-phosphate dehydrogenase. And option 4, phosphoglycerate kinase. So it is actually phosphoglycerate kinase which helps in the ATP synthesis in the process of glycolysis. Next we have chlorosis in leaves is generally due to the deficiency of. So chlorosis, it is actually a fancy name for the yellowing of the leaves. When leaves get older, they'll turn into yellow color, right? The green color changes to yellow color. That process is known as chlorosis. And this chlorosis in leaves is generally caused due to option 1, magnesium and iron. Option 2, calcium and iron. Option 3, chlorine and magnesium. And option 4, calcium and sodium. So this chlorosis is majorly caused due to the deficiency of magnesium and iron, which is the first option and the correct answer. So that's it for the today's class. I hope you all enjoyed and understood everything I taught. And if you have any doubts or any, even the smallest doubt, please don't hesitate to drop down your doubts in the comment section below. And if you liked our video, please like, share and subscribe our channel. Please subscribe our channel and like our video. And please share this video as much as possible with CB method aspirants for more such videos. So keep preparing for your exam. All the best. See you in the next class. Bye bye.